So there you can see clearly. And so the first question was, how did they do this? Is this aggregation? Are these yeasts sort of coming together, or are they using another mechanism? So they could be aggregating like this. This is what some organisms do. Oops, like dictyostelium, a slime mold. It has a free living stage where it lives as a single cell, and then when resources are low in the environment, they'll come together. They'll form this amoeba-like superorganism, and then they make a fruiting body. And distribute the spores. The other way to do it is post-division adhesion. So let me try that again. You can see how they don't ever let go of the mother cell. So when the daughter cells, when it divides, they don't completely let go. So that's post-division adhesion. Theory predicts that post-division adhesion will be more likely because these are more closely genetically related. In fact, they're clones, right? They're the same. So there's dictyostelium with the fruiting body, and here's the way that they come together and aggregate to make the stalk, and then some make the fruiting body with the spores, and then the spores get sort of blown away, and then they start over again. Okay. What you're seeing here is a short video. It's 14 seconds long, and there's a freeze frame. These are the multicellular yeast that are in a drop of water, and they're growing. So one of the other things that you want to see in an individual is division of labor and determinate growth. So these are not aggregations of cells. They're post-division adhesion, and they're only growing to a certain size. Right? Here, let me play that again for you. You can see them. So what we had done here is broken them down with enzymes back to the single cell stage, and then they would grow up into these clusters. And then the clusters, and they're moving together here just by the drying up of the water drops. So they're not motile. That's just a result of the. And that's an overnight. That's a 12-hour time lapse, right? So that's them growing from the juvenile to the adult stage. And the question for us was, are these individuals? Have we evolved multicellular individuals? What do we see here? This is, again, the kind of question that was motivating earlier work on individuality. And this also raises a question about levels of selection, where selection is acting. Is it acting on the individual units, the single cells, or then at some point does it start to act on the group? But when it acts on the group, is the group now becoming an individual? Are you getting a frame shift in the type of individuality from a single-celled individual to a multi-celled individual? So here we have Charles Manning Child. In short, the organic individual appears to be a unit of some sort. Its individuality consists primarily in this unity, and the process of individuation is the process of integration of a mere aggregation into such a unity. For this unity is not simply the unity of a chance aggregation, but one of a very particular kind, a highly constant character for each kind of individual. So we have determinate growth. That was one thing that we were sort of looking for. I'm going to show you another video. And this is a change in reproduction. So here you can see these are single colonies or single individuals, single clusters, and right? Let me try one more time. No, it wasn't going? Let me go back. Sorry about that. It's good. Yeah? Okay. So here, and now what you're going to see is that they reproduce by letting go of a propagule. It's not single cells that break off. They have a juvenile stage. So there's an adult and a juvenile. The blue is adult. Those are twins. Blue and green, adult, juvenile, adult, juvenile. So when they get to a certain size, they start to reproduce these propagules, and then they grow to adult size. What's interesting is, and actually let me skip ahead here, is that we're looking for now in these individuals some kind of division of labor. Different cells doing different things, some kind of differentiation. And we're also here talking about a different kind of reproduction. It's not single cells dividing, rather it's programmed cell death that is creating apoptosis, that is creating these breakpoints where the propagules let off. So what we think is going on is that these apoptotic cells, these orange cells here, serve as breakpoints for the propagules. So you have this, you could have just random breakage, or you could have this programmed cell death, and where those nodes are is where you'll get these, once the cell dies, you'll get offspring, right? And if breakage is random, we would only expect them to break at the apoptotic cell about 5% of the time. And yet we found more than 75% of the time they break at the apoptotic cell. And so it looks like this could be early division of labor, and that would be early differentiation. That would be further evidence that these are not collectives, but they're growing individuals, a new kind of individual. And what we think this does is connect, in a very interesting way, the levels of selections question, the major transitions question, questions about empirical investigation, again, connecting to this project, empirical investigation with sort of a deep conceptual framework. We realized when, those guys actually realized when they first started the project that the empirical result would not be sufficient to show people the phenotype itself would not be sufficient. What was needed was a sort of historical and philosophical framework to explain and put into context the way that the projects do here the empirical result. So I'll just end with this, and again make a pitch for the role of history of science and philosophy of science in these kinds of investigations. My advisor, Fred Churchill, wrote this paper called Darwin and the Historian a few years back, but I think it's important, and it's been really influential in the way I do my work, where he writes, "The fortunes of scientific theories determine the historical reading of the past. More sophisticated histories attempt to fill in the details in the context of science and produce low-level causal theories about network of events. Such efforts require the employment of all the tools available at the historian's bench. For the historian of biology, these efforts demand first and foremost an understanding of the science involved. Without this primary implement, the history of biology was at one time chronology without substance. With it, we have begun to get deeper into the subject." And I think that's the kind of work that I've seen here in these integrated projects, and that's the kind of work that I've tried to do with my team at the University of Minnesota, Mike Travisano, Ford Dennison, Will Ratcliffe, and then undergraduates, Kristen Jacobson, Mitch Hoberman, Amanda Neubauer, Jennifer Pence, and graduate students Jonathan Fankhauser and Chris Orley. So that's all I have for today. Thank you very much.
So we have 20 minutes for questions.
All the way in the back.
Oh, Vitor. Hoje você é baixinho. Não perder peso. Você está no processo de dentro do desaparecimento.
Bom, eu vou falar em português, tudo certo?
I translate. Just keep slow. Fale lentamente.
Certo. Marco, eu queria saber sua opinião com relação a...
Devagar.
Isso é meio devagar.
Nice work. very
No,
slow.
I followed. I followed.
Yeah. I think it's okay for
He
Sean.
wants to know your opinion.
Yeah, I got it.
E o que eu não sei falar em português, the major transitions. Do you think that eu não sei falar major transition em português, né?
I mean,
Okay.
Yeah, the main transition.
Você acha que em todas as major transitions a gente teve um papel importante da seleção de grupo?
The question is opinion about if in all major transitions have group selection played an important role.
I don't think necessarily, and I don't remember all of the transitions that Maynard Smith and Safmari identify. I know for me and for this project, obviously the unicellular or multicellular is one of them, and I know that they have at least one of the major transitions about the evolution of social behavior. But I'm not sure about the other ones, but I would think that it's not the case that group selection would be necessary for all of them, even not remembering what they all are. And in fact, for Maynard Smith and Safmari, they're not necessarily convinced about group selection. Maynard Smith never was. But I think what's interesting, and this was sort of evident in your guys' project, and I think we talked about this a little bit in the lab meeting, was the language is really changing here. People talk less and less about group selection for various reasons, and talk more and more about multi-level selection. And multi-level selection, it seems like even someone like John Maynard Smith might be okay with multi-level selection in some sense. Does that make sense?
If I just add one minor comment, that major transitions have to do with changes in the way information is stored, transmitted, and evolve. And these can have to do something to do with group selection or not, because there are several different ways of changing storage, transmission, and evolution of information. Pedro?
I will ask a very naive question because I don't know anything about this literature, but this experiment that you did, I almost cried here of emotion. You developed artificially a multicellular organism. Is it common? What other
I mean, well, so all right, I convinced you.
I almost cried. I must confess.
Well, I mean, so it was so as soon as they saw those groups, they thought this could be really something very big. They were very excited, and that's when I got the photos slid under my door, and that's when we wrote the grant to continue the work, because they had sort of just done this as a side project. When they were listening to David Queller's talk, they thought, okay, he had this continuum of individuality, where he had these kind of social microbes and then colonial organisms like a Portuguese man-of-war, all the way up through what we take to be totally integrated individuals. And they thought, could we evolve a multicellular individual? Mike Travisano has been doing experimental evolution for 20 plus years. He works mostly with E. coli, but he and Will, who was a grad student at the time, was a PhD candidate, thought that yeast, because it was a eukaryote, because it had sex, because lots of things, that it could be a good candidate. And it worked. It took 4 weeks for them to get that first. And then we went out for beer, and we talked about it, and I thought, wow, this is great. Yeah, well, no, and then, and the initial response was very interesting, because not everyone is convinced that those are individuals, right? And it's not clear what it means to claim that they're individuals, and the kind of language that we used, proto-organism, proto-multicellular, we felt like we had to be very careful, and the review process. I mean, we published the paper in PNAS in January of last year. It was reviewed and sent back to us, 3 times at Nature, twice at Science, and each time they wanted actual mechanisms. They wanted us to do genetic work. There was work that we were doing, but we wanted to get the paper out, so it ended up in PNAS. But a lot of times, for example, one of the complaints that came from a lot of people familiar with yeast is that yeast oh, I'm going to forget the name of the phenomenon. Yeast have this ability when they're in scarce resources the cell elongates, and they make these kind of hair-like structures where they're multicellular, right? And so the first thing, it's called flocculation. So they said, the first thing we heard was like, oh, this is just like a weird flock. It's not multicellularity. But flocculation, the phenotype is totally different, and so we had to make arguments to convince people that this was not flocculation. Other people argued, well, it's possible, or actually there are people who've claimed that yeast was multicellular 750 million years ago. So we just sort of turned on an old genetic switch. It's not de novo. It's possible. We're not making the claim that that genetic machinery wasn't buried in there somewhere, right? But you know, we felt like the phenotype was sufficiently interesting to sort of move forward. And so now we're trying to use selective pressure to see how strong the sort of multicellular characteristic is. Can we devolve them back to single cell state? We're having some success with that if we let them sexually recombine. So, you know, it's a process of convincing ourselves and then trying to convince other people that that's indeed what we have. But I mean, again, we can see historically that this is something that biologists have thought about, and you can read Kant, and Kant talks about the nature of the organism and what it means for an organism, for something to be a biological organismic system. And so they're very organismal to me. I kind of think that they should count. But I think it's going to take a long time to... The thing that's also cool about it, it's a very easy system to work. So we've written up a report for a methodology for doing this in high school labs and undergrad labs. It's a model system that now we can talk about adaptive radiation, we can talk about speciation, we can talk about development. It's a model system that has a lot of potential, you know. And the individuality question is sort of only part of that potential.
Send me the protocol for high school. I'd like to
Yeah.
let the students to do that. And my empirical colleagues, anytime you have a fantastic result, please keep something under my door.
I read the book about individuality and evolution of Julian Huxley a long time ago, so I don't remember exactly what he said there, but I believe that he said that evolution has a tendency of necessarily produce individuals, living beings that are more individuated than others, than before, and he also said that it can be a criteria of progress.
Yeah.
What do you think about these two ideas of Huxley?
So I don't know so much about the first one, about the context of where he's saying that individuation is a natural part of the evolutionary process.
Individuation in the sense that evolution tends to produce living beings that they are not the words of Julian Huxley, but what he said is that evolution tends to produce living beings that are more autonomous in relation with the environment. More autonomous and independent of the contingencies of the environment. And this is a criteria of individuality, it is also a criteria of progress.
Yeah, no, I think, so I think that that's something about sort of the integrated nature of the individual. So the organic individual can sort of buffer environmental effects, right? So it's not totally subject to the extremes of the environment. I remember that, and I think that makes perfect sense, right?
Yes.
In some ways. The question about progress, this was Huxley's thing. I mean, I think he again, here's a good example of someone who saw essentially political and social theory in a biological idea, in a biological theory, And as a representative to the UN for UNESCO and all of those things, I mean, he was somebody who argued that yes, evolution was a progressive process, that it was deeply progressive, and that this would be a natural kind of move up the lineage.
So, but independently of name that progress, you accept that there is a tendency in evolution of producing this kind of high individuation.
No, not necessarily. I mean, I think for both the question of progress and high individuation, I mean, I guess, individuation to some extent you need, if you go back to the origin of life, you need a sort of a semi-permeable membrane to start, right? Maybe you have the replicating RNA out there, but it needs to be at some point enclosed, so it's not just a molecule floating in an environment. It's something that is now somehow self-regulating and then replicating. So that membrane is important, but that membrane is always permeable. So there's always an exchange between the organism and the environment. So there is individuation, but there's not necessarily complexity. I mean, the biomass is still largely single-celled, right? I mean, and I think now we have, so I think that's the best argument against the notion that there's some kind of orthogenetic or even evolutionary trend towards complexity. The random walk, the falling off the abyss, there may be a minimal wall of complexity that we're sort of randomly staggering away from at the single cell level, but I don't think there's a driven trend towards complexity in evolution. I think it's I think it's a random walk. Most things are still very simple. Most things are still single cell.
Mark, thank you for your talk. Part of the controversy around group versus individual selection depends on calculation of fitness, individual fitness, group fitness, and I would like in your example, the Saccharomyces, they are doing both things. Individuals or semi-individuals, if you wish, they are dividing themselves, but the groups are also dividing themselves. So first I would like to hear about the exact example. What do you think could you divide the within group or individual fitness from group fitness or new level of individual fitness? And the second question is just, do you think that, because I have seen many debates around these divisions, and one of the ideas is that, well, you can calculate fitness both ways. If you look at it as a group, if you look at it as an individual, it's just a way of calculating. What do you think about this conclusion?
No, I think that was sort of where the argument had arrived before the argument about the value of group selection, before this kind of major transitions and the revivification with Wilson's 2007 and then the 2010 paper, was that, look, we have mathematically isomorphic models. You like kin selection? Fine. Use a kin selection model, model the phenomena, that's fine. I'm going to use multi-level selection, maybe I use multi-level selection type 1 for this kind of phenomenon of interest, and maybe I'll use multi-level selection type 2 for that one. And I tend to think that that's okay. I tend to think that like a plurality of models in biology is fine. And as long as you have sort of the explicit explanation as to why you choose this model versus that model versus the other model. And I had to ask modelers, mathematical people, like, so, if it's mathematically isomorphic, it means it's the same, right? It's the same, right? Yeah, it's the same. And yet the argument seemed like it was still going on. And they go, why are these people arguing? What is the point of this? I guess the point, right, is if you come to the yeast example, I think it's very hard to say, to say exactly what's going on there, and we're developing models to try to figure out what we think, where selection is acting at what point in the process. So one thing that I didn't mention, but maybe it's obvious to you given that you're a biologist and you understand the experimental setup, but the good thing about these experimental evolution experiments is that you archive the ancestral states all along the way. So then you can take, 10,000 generation multicellular organism and compete it against under varied conditions, generation 10, right? And you can see how they behave, and you can calculate fitness in various ways, and then you can try to figure out, you can try to partition the fitness, or you can, and so we're not there yet. The way I think about it right now is that it's this frame shift. It's a frame shift where selection is initially acting on single-celled individuals to get bigger, and then you get this response of multicellularity, and then this seems like a case where you might not want to call it a group because it's genetically identical. But now the individual is a different kind of thing, and if there's a speciation event, if Saccharomyces cerevisiae is a single-celled and this is a multi-celled thing, I mean, something's happening there, right? I mean, I don't
There's
know.
a development now.
Yeah. Yeah.
One has development and the other has, or
Not.
either don't have or.
Right. The single cell, so microbial people, Mike especially, will always argue like, people think microbes don't have development, but they do.
No.
But certainly, the multicellular looks like it's got development in a way that
I would just try to, the way to calculate fitness depends on what you consider an individual. So,
yeah.
if you
Yeah.
have the evolution of an
Yeah.
individual,
Yeah.
so you will have to change the way of calculating.
Right,
So
that's
it,
right. Yeah, and that's why it's not just a sort of semantic argument for evolutionary biologists. They have to pick. They have to say, this is the important evolutionary unit, and this is an individual, not that that collection, this collection. So Peter Godfrey Smith's book is really good on this: Darwinian population and Darwinian individual.
That's the them.
So is there any, is there any evidence that, or are you looking for anything about that cell that dies that then cleaves those that's similar or different? Could those be mutant cells and therefore aren't the same individual in some sense?
So this this was one of the earliest assays that we did, was staining them with that calfluoride and finding these bright orange, very pretty dead cells, and they looked initially like they might be at the center, so we thought it might be like resource scarcity, like they were getting starved in the middle. But then they started showing up in other parts of the snowflake. And so then we thought maybe it is just size and they get torsioned or something, they just get twisted and that's how they die. But we have a grad student, Jonathan Frankenhauser, who's doing some sequence work and trying to figure out what exactly is going on with them. So yes, it's an area that we're looking at, but we don't have any, we have very preliminary kind of results. I can't really speak to.
In the same text book you quoted, Julian Sorel, 1912, he identified two types of individualities, spatial and individuality in time. Spatial individuality, individuality in time. He said coexistent part and part that succeed each others in time. Did you find this type of, at least individuality in time, can be applied to this experience?
Maybe, yeah, that's an interesting question. Maybe the question of the individual in time could be useful in thinking about the way that we've identified their sort of reproductive process, that you have this growth process that then leads to the breaking off of the propagule, and so maybe over a period of time, I don't know if that time, I think that time is pretty